our next speaker is Jerome Deval, who's a scientist at Alios Pharma, and he'll be talking about how to break a Swiss Army knife, strategies for targeting virus polymerases with small molecules.
All right, so first of all, I'd like to invite Laurent and the organizers of this meeting. I'm very happy to come here today to talk. And as it was mentioned, I'm going to talk about virus polymerases and ways to inhibit them with small molecules. And I like to compare viral polymerases to Swiss Army knives. I'll explain that in a second. Before I get started, I would also like to thank my employer, Alios BioPharma, for giving me the time and permission to come and give this talk. I'm sure most of you haven't heard about Alios BioPharma. There's a good reason for that. First of all, we're a small company, a startup, about 30 people, and we started about less than 3 years ago. Also, before I get deep into the subject, I'd like to mention a few things about nucleic acid polymerization, DNA or RNA polymerization. It's actually a very simple reaction. It consists in the elongation of a nucleic acid using a nucleotide triphosphate to a longer size nucleic acid, pyrophosphate being a co-product of the reaction. So what is really interesting with viral polymerases is that that simple reaction is actually carried by complex proteins. And a few things about their complexity. One is complexity by way of size. I like to give the example of paramyxoviruses, where the catalytic polymerization activity is actually carried by a protein called L protein. L stands for large, often way over 100 kilodalton proteins. Not only they're large in size, but they also tend to form multimers, whether it's dimers or higher oligomer forms. And I'll touch base on some examples with HIV and HCV polymerase. And so they form dimers often, or multimers. They also interact with other proteins, whether they're viral or host proteins. I'll talk a little bit about that as well. One thing that I find really fascinating is that there's a fundamental difference between host polymerases and viral polymerases, is that viral polymerases tend to do a lot of multitasking, and that's why I call them the Swiss Army knives, because for one thing, viruses carry very few proteins for a lot of them. And so each one of those proteins, whether it's a polymerase or another structural or non-structural protein, has to do a lot of things to maintain, in this case, polymerization. And this is different from host polymerases that tend to make those very large multi-protein entities that are very specialized. So as I mentioned, viral polymerases are different in that matter. You have to multitask. In a way, it's like working in a small company. Another thing about viral polymerases that, for personal reason, really fascinate me is that they are a very important target for antiviral therapies. And I'm sure a lot of you are very aware of things that have been advancing in HIV. Today's date, there's 18 drugs that are approved that actually target the polymerase of HIV. And those molecules are actually a major component of the standard of care for HIV patients. So some of the questions that I'm going to try to address today are the following. First of all, well, how do those polymerase inhibitors work, and how were they discovered? And I'm not going to go through each one of them, but there's really some fascinating stories that happened over the last 30 years in the discovery of polymerase inhibitors. The other observation that they are so effective, how does this happen? Why, as antiviral class, are they so potent? And obviously, they're not miracle drugs, so what are the advantages, but also their shortcomings compared to other types of molecules, and also between the different subclasses of polymerase inhibitors. So I'm going to talk a little bit first about structure and function of polymerases. And this is usually one of the very first figures you see, especially from structural biologists, about polymerases. This particular polymerase is HIV reverse transcriptase. People talk about the right-hand shape of polymerases with the thumb, finger, and palm. I'm not a structural biologist, but really honestly, I've never seen really clearly a hand shape. At best, I would say maybe a baseball glove or something like that. But no matter what, you'll see the thumb, the palm, and the fingers. And in this case, it's more complicated. As I mentioned, for HIV, there's another subunit and an additional functional domain. So actually, I started to study polymerases with HIV, and one of the things that is common between HIV and other DNA replicating polymerases is that there's really multiple kinetic steps and conformational changes that are required for polymerization. And without really going too much into the details, you can imagine your APO enzyme binds to the DNA, then it binds to the nucleotide. The DNA and the nucleotide react, and this is the catalytic step. So each one of those steps actually induces a conformational change for the protein. The one thing that sometimes people don't really realize, or maybe you do if you've done enough PCR reaction, is that this is really happens very fast. And there's about 10 to 50, sometimes up to over 100 iterations per seconds. So I mentioned conformational change and also flexibility. This is just to illustrate these changes that happen. The APO enzyme, in this case for HIV, has this sort of collapsed active site. The binding site opens up for the incoming DNA here, and then you have a second conformational change that takes place in order to have the catalytic activity happening. And this is not just HIV. I think it's fair to say that most, and actually all replicative polymerases tend to follow similar features. The thing about HIV, I think that makes it so special, and is the fact that as a retrovirus, the enzyme needs to convert single-stranded RNA into a double-stranded DNA, which will be the molecule that will integrate human genome. And all of this is essentially done by HIV-RT in actually 9 different steps. And for sake of time, I'm not going to go through those steps. But what I would like to mention is that HIV-RT has found a very clever way to hijack the host machinery. In this case, it uses a tRNA a molecule as a primer for DNA synthesis. And another thing that HIV has evolved to do is really do a strand transfer. And in order to do this, you can see this strand here moves to that part of the genome. It relies on RNase H activity, so a completely independent catalytic function compared to the polymerase itself. So I like to also give the example of dengue virus polymerase because it's another protein that has different functional or catalytic activities. So in dengue, you actually see whether within the NS5 gene, 2 catalytic function, one being the methyltransferase that is involved in the capping of messenger RNA, the other one being the catalytic polymerase entity. And what's really great for biochemists, so people who are interested in cell-free assays, is that you can actually chop those parts of the protein and still retain most of the catalytic function. This has been exemplified many times. It's also true for other polymerases. The one thing that is very different from HIV-RT that I just mentioned before is that this RNA polymerase has more like a donut-shaped function. Donut shape. And the reason for that is that it needs to do a reaction that is very unfavorable, which is really the synthesis of the primer. So it's taking 2 nucleotides and making the first phosphodiester bridge. And this is very inefficient catalytically. And in order to accomplish this, the polymerase has really evolved into having a very enclosed active site in order to maintain those 2 molecules in there that probably have a very low binding affinity. So one thing that I really like about dengue polymerase is that, or NS5, is that it makes RNA from scratch. One thing is it makes its own primer that is different from HIV-RT, and it also caps the messenger RNA. But different virus, different story, and you'll see how viruses have evolved in different ways. This is influenza virus replication. And the RNA polymerase, in this case, doesn't bother about making primers or capping the primers. There's this cap snatching activity. So the polymerase itself is formed by 3 subunits. And one of the 3 subunits contains endonuclease activity. And so essentially, it's an RNase function. So what this RNase does is that it actually cleaves a cellular messenger RNA, and this cleaved messenger RNA is used as a primer for the polymerization, the RNA polymerization. And so this is fundamentally different from dengue virus polymerase. So going quickly, just mention the fact that a lot of those proteins that I described are highly flexible proteins, and they carry multiple functions. They've evolved in different ways. The thing that is, I think, really important about the fact that it's so flexible is that this gives a lot of opportunities for inhibition, and this is what I'm going to start to talk about. I'll give different examples about different viruses. So, and one of the first molecules that I think is fair to describe is a cyclovir that has been discovered back in the 70s. I like to tell this story, and this comes from Eric de Klerk's lab in Belgium, where people essentially, cyclovir, so one of the safest and probably most potent herpes virus inhibitors, was actually really discovered, I would say, by serendipity or preparedness. It was originally designed as an inhibitor of adenosine deaminase, and people found by a screening method that it was also an inhibitor of HCV polymerase. So completely independently from the reason for why the molecule was originally synthesized. So when HIV came into the attention of scientists in the 80s, of course they looked at the handful of known antivirals that were already known. This one being one of the few at the time. They tested against HIV. Cyclovir was not active in HIV inhibition. What's really interesting is that medicinal chemists tend to make lots of, I would say, slightly modified molecules. And one of those molecules here that ended up not being inhibitor of herpes simplex ended up actually inhibiting HIV. And this is really where I would say serendipity or the value of really testing many compounds against many viruses, even if there is really a low chance of working, has led to some of the major discoveries in antivirals. And this molecule here, actually the great part about the story is that this molecule is, although it's structurally similar to a cyclovir, inhibits HIV polymerase by a completely different mechanism of action, binds to a different part of the polymerase. And this molecule actually, with a little bit of optimization, led to the first potent allosteric inhibitor of HIV polymerase. So there's a lot of examples like that, or there's a lot of merit and credit that has to be given to people who test compounds against viruses that are not necessarily have a good chance of success, and that's how you found in those two cases very potent and very effective antivirals. One of the things that especially pharmaceutical industry really likes to do, I call the brute force approach, also called high throughput screening, is a way of just banking on this, I would say, serendipity and just saying, well, let's test everything against the virus. So this has been allowed mainly through the advance of robotics and automated workstation, liquid handling. So allowed to screen 1 million, sometimes 1 million plus compounds. And you can do this in cell-based assay or biochemical assay. There's a few considerations. I'm not going to go too much into the details for that, but obviously price is a big factor. How robust your assay is, signal to noise, but there's a lot of other parameters that are taken into consideration. For polymerase, the detection method can be really critical. Most of the polymerase assays for virus polymerases are actually isotope-based radionucleotide incorporation assays. They actually, people hate to use those assays for HTS because nobody really wants to put radioactive isotopes into robots, or especially when you're going to screen 1 million compounds. So I like to give this example of a clever way of looking at polymerase activity in an indirect way. This is very similar to the pyrosequencing technology where people look at coupled reaction, and with 3 enzymes, essentially you can detect light as a tracer for your polymerase activity. And you can look at the loss of light as a way to inhibit your polymerase indirectly. This is just very quickly looking that between the luciferase assay and the radiometric assay, you can have good correlation in the compound inhibition. One of the things that is often asked about how do you discover compounds and how do you advance compounds. This is one example. I also use this polymerase assay to give you, this is from a real experience with dengue polymerase. So you can use this luciferase assay for your original screening of your 1 million plus compounds. But then you often rely on more direct methods. You can imagine that if you inhibit the luciferase, this is not a compound you're interested in developing. And there's a lot of value for running direct assays. And then there's a lot of, and this can take a lot of time, is the orthogonal validation. So you can look at enzyme kinetics. Is your inhibitor competitive, non-competitive, and competitive inhibitor of the polymerase? Can you set up an assay where you can look at direct binding between the small molecule and the protein target? And if you can you actually get a crystal structure of the complex between the protein and your small molecules? People in the pharmaceutical industry really like to rely on those type of assays. Obviously, you would like to also make sure that your polymerase inhibition leads to inhibition of the virus in a cell-based assay. This is not always the case. You can have actually months or years between this part of the validation and getting to compounds that are actually effective in cell-based systems. But if you have cell-based assay, then this opens a lot of opportunities. One of them being resistance selection experiments and reverse genetics. Those are great tools that are available for drug discovery programs, and obviously iteration of making new compounds and going through the whole screening paradigm. So this has been very effective for HCV polymerase, and today there are 4 molecules that are in clinical trials that have been discovered that way. I'm not going to talk too much about where they bind and what they do, but and this has also led to the finding of 4 distinct allosteric sites in a single protein, which is really quite rare for inhibitors. So this is, again, this is not a perfect way of finding molecules, and there are some major limitations with running HTS. First of all, it's very heavy in chemistry. I've seen teams of medicinal chemists. You have 30, sometimes more than 30 chemists that are dedicated on making variation on a single molecule. And a thing about low ligand efficiency, I'll go back in a few slides, but because it's so heavy and testing 1 million compound is also can be pretty slow and obviously costly. Another issue with running those type of assays, especially for biochemical assays, is that, again, observation with polymerase is that you tend to generate a lot of false positives, a lot of compounds that look very good originally and actually will never turn into drugs. And there's some classes of compounds that fall into those categories. They're sometimes referred to promiscuous binders or promiscuous inhibitors. Essentially what they do is that they form those large aggregates and they prevent the enzyme for being free in solution, or sometimes they even partially enfold the enzyme. I'm not going to go too much into this. This has been very well shown by Brian Shorkett at UCSF, and there's a lot of good papers that refer to those unwanted, unspecific type of inhibitions. And this is a nightmare for, I would say, for biochemists or people doing drug discovery, because you can spend a lot of time working on inhibitors that look like this, and that will actually not be able to advance. This is actually an example for an experiment that I did back in 2008, where you can see in blue your inhibition with your compound, and then you add the detergent. And so by adding the detergent, you actually prevent the aggregate from forming, and suddenly the target is not inhibited anymore. So I would say this is a good example of how you can actually narrow and identify some of those promiscuous inhibitors. So maybe the part that I'm actually more interested in is really the rational design, because in a way it's really doing some targeted libraries, and you can actually design cell-free assays. I'm going to touch base a little bit on fragment-based drug discovery. I saw that there's actually a poster describing this. But there's also very simple ways of running cell-based assay using nucleoside analogs. And each one of those strategies actually really proved to be pretty effective. And I'll just take a minute or so to talk about them. So if you remember what I mentioned about HTS, virtually you screen everything you have in your library. Often the molecules look like that. They're not very small in size, so often greater than 300 or 400 Dalton. And in a way, they don't really bind very well to the binding site. They do bind, usually 10 micromolar IC50 or KD, and that's what's called low ligand efficiency. The idea with fragments is to actually start by screening things that are a lot smaller than those typical HTS library compounds. And this is often under 200 or 300 Dalton in size. The great thing about this approach is that if you can actually identify molecules that are this small, then it's a lot easier to build a molecule in a way that actually you carry a higher ligand efficiency. And this has worked in many cases in finding new molecules, sometimes years after HTS were run for a particular target. And so I'll give you an example about how this works. Is your primary screen, you might want to screen something like 5,000 compounds. They're all very small, like I said, they're called fragments. And you have those binding assay. In this case, it's a surface plasmon resonance type of BIACOR experiment. Then you select your compounds that bind. You try to get a crystal structure of your compound with your target. And from the crystal structure, usually chemists are pretty good at growing the compounds and finding pockets within the binding sites that can accommodate bigger compounds with higher binding affinity. And this go into iterations of making new molecules that bind better, getting new crystal structures. So to give you an example, I know a program where people in about 3 years were able to get over 100 crystal structures of compound plus inhibitors. So you can go very quickly in those iterations. This is a real example from HCV polymerase, again illustrating the small molecules, originally low binding affinity, 1 millimolar KD. Does not inhibit the enzyme, and that's actually something that sometimes people have a hard time understanding, is that those molecules are so small, they don't actually show any phenotype, but they do bind. But the great thing about them is that it's actually fairly easy to expand them, and suddenly you have a submicromolar inhibitor that is also a potent inhibitor of the enzyme. Now, one of the, I would say, good tracking record for antivirals is nucleoside analogs or chain terminators. AZT being the first one identified for HIV. It's a toxic molecule, and I'll touch base on that very briefly. The idea behind is very simple, and actually, my apologies because I made a mistake. There's no hydroxy here. But for those of you who remember the Sanger reaction, or have seen or done some of those sequencing gels, it's very simple. You use a chain terminator, so a molecule that doesn't have a 3' hydroxy, and you can actually chain terminate in a base-specific way. In a way, AZT really works the same way, except this is not Taq polymerase or one of those highly replicative polymerases. This is a chain terminator of HIV polymerase. So another example with dengue polymerase, how do you demonstrate a chain terminator? It's actually very simple. If you have those high-resolution sequencing gel, you can see the extension of a primer from a size, let's say in this case, 18 mer to 19 mer. Then you have the next nucleotide, in this case, CTP, goes from 19 to 20 mer. In the presence of your chain terminator, the polymerase will never be able to convert from 19 to 20 mer. Will stop at the first position, whether or not you have the next correct nucleotide. One of the, I would say probably to this date, one of the most exciting part of the HCV drug discovery and development process is within nucleoside analogs for anti-HCV therapy. So what you can imagine is that those nucleosides have to penetrate the cells, then they convert it to triphosphate by cellular kinases, and then it's only the triphosphate that inhibits the HCV virus at the polymerase site by chain termination again. And something that always, I think, fascinates me is that you can make very, very subtle changes in your molecules. So this is a natural ribonucleoside. This is a substrate for all human RNA polymerases. You make a very small change, you add a methyl, and suddenly you have a potent inhibitor of HCV polymerase. This has gone into the clinic, and there's still derivatives of these molecules that are being tested. Another company has chosen a different strategy where at the fluoro group at the 2' position, which is a very toxic molecule that has been derived to do anti-cancer, can be modified by adding, again, one simple methyl here at the 2' position. And here you have one of the probably the most potent HCV inhibitors to date. And those families of molecules have been heavily patented, and there's a lot of interest to continue to develop such molecules. This is a presentation from the last company that I mentioned, Pharmaset, last month in Germany, where people have seen this very impressive decline in virus load in HCV-infected patients. So the molecules were given only for 14 days in HCV patients. And after 14 days, 15 out of 16 patients achieved an undetectable RNA level in this case, they combine two different nucleoside analogs following this particular chemotype here. Why are they so active? One of the reasons is that I mentioned the chain terminators. I mean, they don't interact necessarily covalently with the polymerase, but actually interact in an irreversible way with the RNA. And they actually compromise the integrity of the viral genome. Only one incorporation per replication cycle is enough to actually stop virus replication. Most of you know, and I took the example from AZT, is that they can be also very toxic molecules. The mitochondrial DNA pol gamma is one of the host polymerases that often react with those deoxynucleoside analogs, especially with AZT, and this has led to very severe side effects and toxicity with those classes of molecules. They're also very difficult. I work with a lot of chemists, and they also complain very much about how difficult those molecules are to make. And I'll skip quickly the advantages. One of the things that I wanted to mention, I think has a lot of potential moving forward with those molecules, is that nucleoside analogs tend to be active against broad virus targets. And there are two publications that I like to refer to. One showing a molecule that is active against both HCV, dengue, yellow fever, as well as other flaviviridase. And this is done in antiviral in vitro assay, but people in Utah have also used small animal models, in this case yellow fever, to show that even in animal models you can see an anti-HCV molecule that is very effective against yellow fever. General conclusion, as I mentioned, polymerases play an essential in virus replication cycle. They are an important target for antiviral treatment. From the past experience we know with HIV, I think in the future HCV will probably be heavily relying on polymerase inhibitors. Some of the things I haven't talked about is how those molecules are used, but one thing that is very true, and this is probably to date the most potent HIV drug, which is a combination of actually 3 polymerase inhibitors called a tripla. And with this, I'll be happy to take any questions.
Somewhat of a philosophical comment. You're in a small company.
Yes.
So you can't afford to do these high throughput, maybe you can't. I mean, in other words, the cost of pharmaceuticals is huge.
Correct.
Most of that cost is the clinical trials.
Yes.
But also it is also in discovery. And so I would like your opinion as to what is the best approach. You mentioned high throughput selective molecular structure, which takes a good crystal structure.
Yeah.
How do you see it as a small company?
Well, actually, I'm really a big advocate of rational design, and especially for smaller companies or even universities. I think I showed 2 examples of either working on selected libraries like nucleoside analogs. It's a very, I would say, specific and sometimes challenging chemistry in terms of medicinal chemistry. The molecules are very difficult to make. If you have the right people that have the right expertise, you can make very few compounds and very quickly get to drug-like properties. This is one example. Fragment-based drug discovery, you can easily buy a collection of 5,000 molecules. Anybody can do that, even small companies. Sometimes a lot of universities carry those libraries, and you can test them against your targets in binding assays, and you can come up with very interesting molecules. You don't have to have this larger screening libraries that big pharma typically have.
I have a question. Have you seen examples of compounds that function by disrupting oligomerization?
Let me see. Okay. So the question about compounds that might prevent protein-protein interaction or multimerization of proteins, you'll find examples in the literature. For small molecules, it tends to be very challenging. By law of mass action, it's very difficult for small molecules to interact with wide surfaces of protein-protein interaction. Molecules that advance far, I'm not aware of.
Final question in the front here.
So you're talk highlighted the differences between viral polymerases structurally.
Yes.
And yet at the end you argued that,
Yes.
that polymerase inhibitors might be sort of pan-viral inhibitors.
Mm-hmm.
So there seems to be a contradiction there because very small changes in the active site would reduce binding sufficiently that you would lose activity.
Yeah.
So how do you reconcile those two different concepts?
So if you, let's say if you do sequence alignment or protein alignment between different classes of viral polymerases or between different hosts versus viral polymerases, you'll most often you'll see very little similarity. The one thing that is very conserved is the catalytic site. And they all carry the same reaction. It's all, they all carry the same phosphoryltransferase reaction. And in that way, this is really one of the reasons why the advantage of this is that you have those broad spectrum type of antiviral. I would say the flip coin is that you also have often some lack of selectivity between virals and the host polymerases. The analogy that you can take is kinases that also are targeted with small molecules. A lot of them are active site binders, and it's very difficult to develop and to discover a molecule that is very specific of one kinase. They tend to be broad spectrum molecules. Even though if you look at the all kinase sequences, they have very little in common if you go among the different classes of kinases. Does that answer your question?